Alright, ladies and gentlemen, so in this last screencast for Unit 5.5 Protein Synthesis, we're going to talk about the relationship between genes and proteins. So, genes are nothing more than instructions for building proteins, like blueprints, really. And all of these things that you see pictured here, the baby, the blood cells, and the flowers, are all comprised of genes at their genetic base, in their DNA. So what do proteins have to do with these, right? What's the relationship? We should already kind of have an idea of this. And the answer is everything, right? Because biology is DNA to RNA to proteins. So DNA and proteins, DNA and genes are kind of synonyms here. DNA and proteins are closely linked in biology. The traits that any organism is going to express is the result of the proteins being made within your cells via transcription and translation. Now let's talk about mutations. From time to time, your cells are going to make mistakes in copying their DNA. It's not a perfect process. An incorrect nitrogen base, like an A instead of a G or a C instead of an A, may be inserted or skipped altogether. And these mistakes are called mutations. Mutations are just changes in the genetic material of a given cell. Now, mutations can either be a gene mutation or an entire chromosomal mutation. Like maybe the entire chromosome is missing, or maybe part of the chromosome is missing. Or maybe the chromosome has many mutations within it. Or you can have a gene mutation, which is a single stretch of DNA, which is what a gene is, and there's a single or multiple nitrogen bases skipped or misplaced within that gene. So gene mutations produces change within a single gene. Chromosome mutations produce changes in a whole chromosome. We can look here at point mutations, right? This is where we have one nitrogen base that's wrong. So we have our DNA up top in the blue, our mRNA that's going to be made from it in the orange, and then our amino acid sequences, which are our proteins, down below. So point mutations are changes in just one base pair of a gene. These are called point mutations because they occur at a single point within the DNA sequence. You can see here, this is the normal unmutated version, DNA, mRNA, and then the amino acid. And then we replace this middle T with an A, that's the point mutation. And now the mRNA is different, and now even the amino acid is different. And believe it or not, this is actually the mutation that causes sickle cell anemia. Now, there are two types of point mutations. You can have base pair substitutions, which is what we have here, or you can have base pair insertions or deletions. Insertions and deletions are very bad because they can cause downstream problems in the DNA. So a base pair substitution, hang on, let's pull all of this stuff in. Hang on, I don't know when it's going to come up here. Looks like we're going to put the whole slideshow in. So a base pair substitution is the replacement of one nitrogen base with another. Like on the last slide, we replaced that T with an A. It would just affect one amino acid, but there's a characteristic called the wobble effect, where if the mutation, a point mutation, a substitution here, happens at the third position in the codon, there's a chance the mutation will be silent. It'll have no effect at all. The codon can still call for alanine, the correct amino acid, right? Because you see here all of these, they're all GC, but then UCAG, it can be any of these last four letters, and you'll still get alanine. So that's a silent substitution mutation. Now, if the substitution were in the first or second position, it would have a greater effect and change the amino acid altogether, which ultimately changes the protein. Now, a switched amino acid may or may not have an effect on the proper function of a protein. But like if it were in the active site of an enzyme, right, that could cause a real problem because that enzyme could be non-functional then because the active site is just one amino acid off. Now, let's look at one more example. So what amino acid would be called for by the codon AAG? Take out your codon chart right now, AAG. The answer is lysine. Now what would happen if an A was substituted in the third position? So it would be AAA. So the codon AA would still call for lysine. There'd be no effect on the protein being made, and this would be a silent mutation. But what if we put a C in the third position of this codon? Now we're going to get asparagine, and the protein is altered. Now what would happen if there was a substitution at the first or second position in this AAG codon? The wrong amino acid will definitely be called for, and the protein being made will be altered. Now, insertions and deletions are a little different. Let's bring all of this onto the screen here. Insertions and deletions are adding or taking away a nitrogen base, and these have disastrous effects. Remember our sentence analogy, the red dog ate the cat, and we removed the first T, so we deleted the T and it shifted everything? That's exactly what happens here. So if the G is deleted in the first codon, so the G here is deleted, the codons will read as follows. And you can see none of the codons are the same anymore, meaning this is going to be a completely different protein with maybe no effect or a completely disastrous effect that the cell never intended. Now the codons are still read in groups of 3, right, up until the end where there is no third base. But you notice all of the nucleotides downstream from this G are different. They're all shifted now because the ribosome is going to read them in their series of 3 like it's built to do in the codons, but we're missing one, and that's going to shift everything to the left. These are improperly grouped codons. So it basically alters the entire reading frame, if you will. And these are called frame shift mutations because everything's been shifted one way or the other. The reading frame has shifted. And frame shift mutations can and usually are pretty disastrous for the protein being made. Now, most gene mutations are neutral and they have no real side effects. Some mutations can cause dramatic changes that normal cell functions are disrupted and may result in genetic disorders, and that happens all the time. Then some mutations may actually be beneficial, and we call them adaptations, and makes an organism better for its environment. Like the mutations that cause these different beaks on finches, okay, which we're going to talk all about in Unit 6 Evolution. And these organisms that have these adaptations, these positive mutations, are better suited and more likely to survive and will be selected via the mechanism of natural selection, which is all coming up next, folks. See you in the next one.